So then we started a 7-year plan on looking at mitochondrial substrate level phosphorylation under different conditions. And this paper is the culmination of all of the research that we did, proving that mitochondrial substrate level phosphorylation is a major source of ATP and energy driving the dysregulated growth of these brain tumors. This is why when you go in vivo now, you can manage cancer by taking away the 2 fermentable fuels. And we plan to do, if we can get funding, we plan to go through all the major cancers: lung cancer, colon cancer, bladder cancer, breast cancer, because they all show lactic acid and succinic acid as waste products. All of these cancers are telling us the same thing. We are fermenting. You want to kill us, you got to take away the fermentable fuels. The cancer cells have been telling us this for decades, since the time of Warburg. But just it hasn't fallen on the ear because everybody thinks it's being driven by genetic mutations. The mutations are an effect of all this process. They're not the drivers, they're the effects.
In a groundbreaking shift from the traditional kill or be killed approach to cancer treatment, researchers are making headway with new strategies that focus on repairing cancer cells rather than destroying them. These advancements have the potential to revolutionize the way we approach cancer therapies and dramatically change the lives of millions of patients. Let's start with a fascinating development from Stanford Medicine. Researchers there have engineered a novel molecule that forces cancer cells to self-destruct using a process known as apoptosis. Here's the twist. They've combined 2 proteins, BCL6 and CDK9, to essentially flip the survival mechanism of cancer cells on its head. This new method targets the very genes that would otherwise keep cancer cells alive, forcing them to die instead. Specifically, this molecule could be a game changer for cancers like diffuse large B-cell lymphoma, which has proven difficult to treat. What makes this approach even more promising is its precision. Early lab tests show that it effectively kills cancer cells without harming healthy tissue, unlike the collateral damage caused by chemotherapy or radiation. And while it's still in the testing phase with live animal models, the early results suggest that this method could minimize side effects and offer a much more targeted treatment. Meanwhile, across the globe at the Korea Advanced Institute of Science and Technology, KAIST, scientists have pioneered another bold approach. They've figured out how to revert colon cancer cells to a state that more closely resembles normal cells, all without killing them. This technique taps into the natural differentiation processes that govern normal cells, allowing researchers to switch cancerous cells back to their more benign counterparts. This breakthrough isn't just about reversing cancer. It also addresses one of the major issues in cancer treatment: resistance. By restoring cancer cells to a normal state, this method offers hope for therapies that could be effective over the long term without the harsh side effects that plague traditional treatments. Together, these studies represent a monumental shift in how we think about cancer. Rather than focusing on the brute force of destruction, scientists are now turning to innovative ways to repair and revert cancer cells. And while the research is still in its early stages, the implications for future cancer therapies are undeniably promising. A future where cancer doesn't have to mean destruction, only repair, may not be as far off as we once thought.
So where are we at? We've done enough work on these chemicals that we've now identified sets of them and even a single chemical that we're treating mice with. We know already that they're safe in vivo, and we're going for it. We're going to see if we can reverse senescence in vivo, cellular senescence, and hopefully lifespan will be improved, and perhaps health along with it. And that'll probably be for next year. But that's where we're at. So what I want to emphasize is that what seems to be apparent from work from other labs like Vittorio's and Juan Carlos Belmonte's lab, many others, Tom Rano, we are seeing that reprogramming can restore identity of cells from many years before, allowing them to behave like they were young again, quite literally because they are young again. And this is an example of that shown graphically where over time during aging, cells lose their identity, becoming this red ball, And Waddington would be proud. We now seem to be able to push the balls back up, back to where they came from, even if they're cancer cells in some cases.